Okay, let's get into the good stuff. How does that delicious food that you ate earlier today turn into energy? Whether we are talking about a person, an algae, or a bacteria, somehow you have to have some sort of energy source, some sort of often a food source that can be broken down to give you energy. We are going to mainly talk about glucose, a type of carbohydrate, because glucose is the most common energy source for microbes. So we're talking about carbohydrate, because glucose is a carbohydrate, catabolism, because we're breaking down a molecule. In this process, there are going to be 3 different stages: glycolysis, Krebs cycle, and the electron transport chain. I want you to repeat that with me because you need to be able to tell me that. The 3 processes of carbohydrate catabolism. Say it with me: glycolysis, the Krebs cycle, and the electron transport chain. Okay, the first step in glycolysis, you are going to take a molecule of glucose and split it in half. Okay, so glyco meaning sugar, lysis meaning splitting. So 1 glucose is going to turn into 2 pyruvic acid. Glucose is giving up oxygen, so it is being oxidized, so pyruvic acid is more oxidized. Okay, you with me so far? When we do that, NADH picks up those electrons. So we are going to make 2 NADH. Okay? Then we are going to take a little bit of energy. Takes money to make money, takes energy to make energy. We're going to first use a little bit of ATP. We're going to burn 2 ATP. Then we're going to make some energy. We're going to make 4 ATP. So altogether, we spent 2 but made 4. We have just made 2 ATP. That's what you need to know about glycolysis. So if you have that chart in front of you, you can go ahead and fill that in. For glycolysis, where it happens is in the cytoplasm. For a eukaryotic cell or a prokaryotic cell, this happens in the cytoplasm. What type of energy you're making? You are making 2 NADH. Those are carrying electrons that are going to go to the electron transport chain. You made 2 ATP. You netted 2 ATP. That's how much energy is going to be available for your cell right now. Then the other important things that happen in this step is 1 molecule of glucose is oxidized to 2 molecules of pyruvic acid. Okay, so here's kind of the whole process. If you are a biology major, you have to look forward to memorizing this table eventually. But you burn 2 molecules of ATP, the energy using stage. So you burn 2 ATP, and then you make 4 ATP. So out of that, you have just made 2 ATP net. You start with glucose. This is your glucose, these 6 balls together. You end with 2 pyruvic acid. That's these 3 balls together. And then here at this step, we have made 2 NADH. Do you remember what NADH is doing? It's carrying electrons. Okay, what is NADH doing? It's carrying electrons. We made 2 NADH. It's going to carry electrons to the electron transport chain. Okay, now what's going to happen to our pyruvic acid? It can go through 2 different ways. It can go through respiration. Respiration is the best thing to do because it makes tons of ATP. It's going to oxidize that pyruvic acid all the way to carbon dioxide. And at the end, those electrons will be picked up by something else, oxygen or some other inorganic molecule. If for some reason it cannot go through respiration, it'll go through fermentation. In fermentation, pyruvic acid will not be completely broken down. It will only make low levels of ATP, but it's enough to keep that cell alive until it's able to go through respiration again. So if it is going through, we're just going to assume that it is going through respiration. The next thing it needs to do is through a preparatory step or an intermediate step. Depending on what reference you're looking at, it might call it some different names. I'm going to call it the prep step or the preparatory step. In the preparatory step, pyruvic acid is going to be oxidized. Oxidized means it'll give up electrons. So here you can see it's giving up electrons, creating 2 more NADH. What is NADH doing? Carrying electrons to the electron transport chain. Very nice. It's also going to be decarboxylated. That means it's giving off a CO2. So at this point, if you're something like a person, you breathe out that CO2. It just breathes right out through your breath. With that CO2 now, we've just knocked off one of these blue balls. So you have 2 balls of acetyl-CoA. That acetyl-CoA is ready to go to the Krebs cycle. So if you are filling in your chart, this is happening in a prokaryote. It is happening in the cytoplasm because that's what it's got. In a eukaryote, it is now moved into the mitochondria. The mitochondria is where most of these metabolic reactions happen in a eukaryotic organism. What's important here, we made 2 NADH, which is going to carry electrons to the electron transport chain. We made 0 ATP. And the important other details to know there is 1 molecule of pyruvic acid with its 3 carbons is chopped down to a 2-carbon acetyl-CoA. We have 2 pyruvic acids, 2 acetyl-CoAs. And that decarboxylated means we just knocked off this carbon dioxide, which can just be breathed out as a waste product. The next step is the Krebs cycle. And this looks a little bit busy, but it's really not that bad. So we take 1 molecule of acetyl-CoA and we are breaking it all the way down to carbon dioxide at this point. So we are releasing those 2 carbon dioxides, 1 here, 1 here. Okay. Because there are 2 acetyl-CoAs, we're running through the cycle twice. So after 2 cycles, we have made 6 NADH. 3 for the first acetyl-CoA, 3 for the second acetyl-CoA. We have made 2 molecules of FADH2. Ooh, there's a new guy. So remember what we said NADH is doing? It's carrying electrons to the electron transport chain. Guess what FADH2 is doing? It's carrying electrons to the electron transport chain. We've also made 2 ATP, 1 for the first cycle, 1 for the second cycle. So 2 ATP. So now, that sugar that we originally started with has been broken all the way down to carbon dioxide. We took electrons out of it, and those electrons are being held in NADH and FADH2. So most of that energy that we originally had in glucose is now held in those electrons of NADH and FADH2. We've made a little bit of ATP. We made some ATP in glycolysis and a little bit of ATP in Krebs cycle. But most of that energy now is in NADH and FADH2. NADH and FADH2 are holding electrons. They're taking those electrons to the electron transport chain. In the electron transport chain, all of those electrons are going to be used for their energy to give you lots of energy. Okay, so now you should be able to tell me what happens during, let's review this also. Krebs cycle, back to your chart. Krebs cycle is occurring within the mitochondria of a eukaryotic cell. In a prokaryote, they don't have organelles. It's happening in the cytoplasm. In the Krebs cycle, we have made 2 different types of electron carriers, 6 NADH, 2 FADH2, 2 ATP. Put that in your chart. In the Krebs cycle, acetyl-CoA is broken all the way down to CO2. So 2 acetyl-CoA turn into 4 CO2. That's what you need to know there. Okay, so now you can tell me what happens during glycolysis. Where does it occur? What is decarboxylation? What are the products of the Krebs cycle? Your little chart, you have it filled out for glycolysis, the prep step, and the Krebs cycle. For each of those, you can tell me where does it happen in a prokaryotic cell? Where does it happen in a eukaryotic cell? You can tell me how much ATP is made in each step. You can tell me how much electron carriers are made in each step. and you can tell me what is actually going on in that step. Okay, next is the big, fabulous electron transport chain. I will meet you back for our next video. My favorite thing to talk about, because I don't know, maybe I just like eating that much, but I love to talk about the electron transport chain. I'll meet you back in our next video. Bye.